Okay, in this video we're going to talk about condensation polymerization. First of all, we've got to make sure we understand what we mean by condensation reaction. So I'll do that before talking about condensation polymerization. And then recognize that we can have a number of different types of condensation polymerization. So we can have polyesters and polyamides, and both of those can either be made from the same monomer repeated over and over, or from alternating monomers, and I'll explain all of that. It's important to realise that all biological polymers, and you know what they are from your biology studies, come from condensation polymerization reactions which are mediated by enzymes, and that we can, and particularly in biology, can reverse the condensation polymerization by hydrolysis. All right, so we're going to look at all of that in a little bit of detail. Let's start off by making sure we know what we mean by condensation reaction. So in a condensation reaction, we would take either an alcohol, say ethanol, and react it with the carboxylic acid. So ethanoic acid, we've got this carboxyl functional group and this hydroxyl functional group. If you react those together in a little bit of acid, concentrated acid, and heat them up, you'll find that you can persuade it to undergo a condensation reaction. And in all condensation reactions, you end up losing a molecule of, well, a small molecule, usually water. And then what happens is the OH from one of those two molecules, and we know in chemistry it's usually the OH from the carboxylic acid for reasons that I'll spare you. And how we know I'll spare you as well for now. And the H from the other molecule, which is usually the H from the hydroxyl. If you get it wrong and it's the other way around, it doesn't matter, you get the same, it's the same outcome. So you end up forming a water molecule from the H from one of the molecules and the OH from another. You end up with water there. And what happens is the oxygen now has only got one bond, so that clips, this ends up clipping to that carbon there, making an ester, right? Oops, dear. There we go, CH3, CH2, O, and then it's clipped to that carboxyl group minus the OH. Okay, so we've ended up, that oxygen bonded to that carbon there, making an ester linkage. You could do exactly the same idea. Instead of alcohol, you could use an amine. I'm going to use methylamine or methylamine. And although you wouldn't do this reaction in the lab in this way, I'm not going to talk about why. Suffice to say, this is a base and this is an acid, so you get problems there. But in theory, at least, you could take an H off here, so one of these two hydrogens, and the OH from over here, and clip it together. And this can be done in biology at least, but not in a lab. If you lost one of the hydrogens, and then that N is connected to the carbon of the carboxyl, and we end up like that, plus H2O. So we end up with an amide in this case. So that's an amide. Don't get, it's one thing to make sure you get your heads around, that's an amine, and that's an amide. Amines can be primary, secondary, tertiary. They're basic, they're nucleophilic. Amides, not basic, not nucleophilic, and have got a D in them. You can also do condensation reactions. Instead of an OH here, this could be a Cl. So you could have an acyl chloride and you can end up forming HCl here. Little minor extra details for you that you don't really need, if I'm honest. But you can do those kind of reactions. So condensation reactions, formation of a small molecule, typically water, from an alcohol or an amine, together with a carboxylic acid or an activated carboxylic acid like an acyl group. All right. Okay, so what about, instead of having a simple alcohol, how about we have a di-alcohol, or what we call a diol, and there's the diol, and react that with a dicarboxylic acid or a diacid. And sometimes you see these things drawn with a kind of blob representing whatever's between either end of this doubly substituted molecule or doubly functionalized molecule. It could be a circle or a square or a diamond or a star or something, and likewise here. But I like to do it with proper, some sort of proper structure that could exist, right? And does exist. You can combine these two together in exactly the way we were just talking about, and we lose the hydrogen from one molecule and the OH from another, and they combine together to give an ester. This isn't a polymer yet. All it is quite a large, by IB standards, molecule that's an ester. And of course that would generate water as a byproduct. There we go. So that's the first stage of a condensation reaction. And you can see that we've got an ester linkage. Now, you've still got on this molecule two functional groups. You've got a hydroxyl on one end, and that can react again, just like this one did, with another carboxylic acid over here. So we'll end up with carboxylic acid, or dicarboxylic acid, dialcohol. Dicarboxylic acid, dialcohol. And likewise the other way, this carboxylic acid can end up extending this way. So we can then react with an alcohol, then a carboxylic acid, and so on and so on and so on. So you just see that they extend onwards. And the way that we draw that is to draw essentially the same thing again, but remember to lose another molecule of water from the outside. The H from the alcohol, from the hydroxyl, and the OH from the from the carboxylic acid here. And then we draw that with a repeating structure like so. And I'm not going to go through it. You can do exactly the same thing with diamine as well. So it'll be NH there and NH there. But look, we end up with the O here. We've got rid of that H. We've just got an overhanging bond over a square bracket. We've got rid of the OH on this side. So it's showing you that this C double bond O here is bonded to, or this carbon particularly is bonded to the oxygen that looks like this. And this is the thing that's repeated over and over. And it makes sense because we've got one ester in the middle and one ester on the outside. And that helps you to understand why we must therefore, for every repeat unit, have two N lots of H2O. If you're being very particular about it, of course, these chains are finite. It might be 100 units long, let's say, or 1,000 units long, and you end up with an H and an OH on either end. And so really it's 2N, and then it's not 2N minus 1, but yes, so 2N minus 1 H2O. 2N and then minus 1, lots of H2O in the end. Right, I think you'd probably get away with just saying 2N H2O. And I'm going to show you examples of that. I'm not going to draw these out. I'm going to show you from the Oxford textbook. We bought a load of them. You can have a look at this if you want to. But here's the Oxford textbook giving you an example. So this is basically what I just talked you through here, just to show you, it's showing you as 2NH2O there. Okay, here's an example of nylon forming, where we've got not a dialcohol this time, but a diamine, and the H is lost, and the OH is lost, and they clip together, then the OH is lost, and the H is lost, and the H is lost, and the OH is lost, and you end up with this. And this is just showing you the repeating unit, and I don't know how well you can see it on the screen, but this carbon to this nitrogen is the repeating unit, and so you'd end up drawing it, I shouldn't really write on this, like that, like so. Okay. You can also get polymers from the same monomer if it's got a different functional group at either end. So I'm going to use, for variety, I'm going to use an amino acid on this occasion. So obviously amino acids are important in biology, and I'm going to use glycine because it's the simplest amino acid. And although you couldn't do this reaction straightforwardly in the lab, biology can do this reaction very well. And so just imagine a whole load of glycine molecules. The H, just like it would have been for the alcohol or the hydroxyl on the previous page, the H of the NH is gone and the OH of the carboxyl is gone. We end up drawing in the repeat unit like so. So NH, NH, C, H, H, that's those ones, and then C, O, and then the square brackets. And just like before, we end up with a molecule of water Now, because there's only one molecule of water required to be lost to form one of these, there's no central. It's not a double molecule to make a repeat unit. It's one molecule. You've got one molecule of water per repeat unit. So you could write N minus 1 H2O because of the OH on the H2 on the outside, if you like. But again, that's a fairly picky point. As I was saying before, it's worth considering that biological molecules break down with hydrolysis, and all that is if I give you an example of, I'll show you from here. I'll show you here. It's an example of hydrolysis from the textbook. So this time it's showing you how a disaccharide has two sugars linked together. You don't need to know the details of this, but you can see that we've got an H and an OH, just like before, from two sugars. They click together to make what they call a glycosidic bond. Don't worry about the name, and a molecule of water. And the reverse process of that is adding water to break that bond, and you end up back here again. It's just showing you that hydrolysis and condensation are the reverse processes of each other. Very, very important in biology. So, for example, we need to go from amino acids to proteins. You go through condensation reactions, or your body makes condensation reactions. But then if you want to break down, you eat some protein, let's say, you want to break it, your body wants to break it down into amino acids, which can be absorbed and then made into new proteins. It would take those amino acids, it would form those amino acids from breaking down the protein via hydrolysis. And that might be done under enzymatic conditions and helped by different pHs and so on. Okay. Hopefully that's what you need to know for condensation polymerization.